Hello everyone, your host Daniel Hutchinson here for the Intractable Polymer channel. Here stands a eucalyptus tree in my backyard in the state of Arizona, one of many different transplanted species that can be found here in my home state. In this video, I'm going to extract genomic DNA from my tree. To start with, I'll need a half a teaspoon of your everyday household table salt, sodium chloride. Sodium chloride has two important functions. First, it removes proteins from cells and DNA and keeps these proteins dissolved in the aqueous layer so they don't precipitate in the alcohol, which we're going to use later, along with the DNA. Second, the sodium ions create a temporary attraction between the positive sodium and the negative phosphate backbone of the DNA, thus neutralizing the DNA, thus making DNA more hydrophobic and can be easily disassociated or precipitated from the water. Here, a sample from my eucalyptus tree. The leaves have been plucked from the stem and cut with scissors into small pieces, making for an easier grinding up for the mortar and pestle. A little bit of cold water is added to aid in the process of breaking down the leaves. About 100 milliliters of cold water is readied for a mixture to work with. With the mixture ready, it's time for its filtration. The readied filtrate is placed back in a beaker. Then, a stable spoon of powdered detergent is added. Mixing is done slowly and easily. Now we let it stand for about 10 minutes. Detergent molecules dissolve the cell membrane of the plant cells by interacting with the lipid particles within the cell membrane. This exposes the DNA. After allowing the mixture to set, it is poured into a test tube, filling the test tube to about a third full. Then 5 milliliters of pineapple juice is added. The mixture is shaken for the pineapple juice to do its job. Again, the mixture is made to set for 10 minutes. These are protease enzymes and are able to break down proteins bound to the DNA. The test tube is now held at an angle, and 5 milliliters of ice-cold ethanol alcohol is added to the mixture. The ethanol flowing down the wall of the test tube forms an interface between the two liquids within the test tube. The plant DNA will precipitate within the ethanol alcohol in the form of a white-colored gelatinous filaments and clumps. The alcohol is a nonpolar solvent. It makes the sodium DNA bond even stronger. So DNA becomes even more hydrophobic and eventually precipitates out into the ethanol layer. Now the top layer of the extracted DNA within the ethanol is eyedroppered into a separate test tube. The contents of the test tube is poured into a beaker, and a sample is eyedroppered onto a microscope slide. Here we go. Let's take a look. Lumps and clumps of genomic DNA can be seen within the sample. DNA, the building blocks of life. Contemplating on a broader scale, DNA can be extracted by the method shown from a practically infinite variety of plants within my own geographical location. I hope you've enjoyed this video, and thank you for watching.